can understand the power of molecules for the thousands of reactions in biochemistry, but most of those reactions largely occur outside our everyday awareness. Even hormones like insulin and epinephrine, which exert effects that we can feel, are spread over a long time, and the effects are often subtle. In this lecture, we'll turn to the most important group of biochemical signals that we do notice, the molecular reactions that give rise to our five senses. These are the signals that give us virtually all our conscious experiences. But just like a painter brings a painting to life using only a brush and some paint, our brains color the canvas of our world, blending signals from our senses. The receptors for sights, sounds, tastes, smells, and feel offer distinct inputs that we assemble, interpret, and respond to. Whether we fight or take flight, love or loathe, stand tall or stand back, ultimately comes from the input of our senses and the processing of our brain. Decision-making in the brain is a kind of neural activity that differs for the most part from the neural signaling coming from our senses. Brains use an elaborate if-then logic to interpret and make decisions about all that input, like a computer program, but with more inputs to consider. Neurons suited to this kind of if-then processing are multipolar. Each neuron can take input from several dendrites and then pass one signal through its axon, which may itself have multiple connections. Sensory signaling, by contrast, works better with simple yes-no messages, signaling when a photon of light is detected, for example, or otherwise resting. Neurons performing this kind of signaling are called bipolar. The signal arrives from a single dendrite and leaves through a single axon. The process happens without making any decisions. Okay, let's consider our senses, starting with taste. We detect 5 kinds of taste: sweet, salty, sour, bitter, and savory. The savory taste is sometimes called umami, derived from a Japanese word meaning delicious. Sweet tastes signal detection of energy-rich carbohydrates, which is why we are drawn to sweet food. Salty tastes encourage us to obtain the sodium and potassium necessary to maintain water and salt balances. Sour tastes may indicate spoilage for detecting foods past their prime. And many poisonous substances are bitter, so detecting bitter tastes could be life-saving. Umami gets its savory taste from the amino acids glutamate and aspartate, alerting us to the nutrients in protein-rich foods. There's evidence we can sense fats due to how they feel in our mouth. In addition, there's also evidence of taste detection for fatty acids, with a surprising purpose. Like sour and bitter, this more recently investigated taste of fatty acids is unpleasant. The sense appears to be a warning sign against fats that have started to break down and go bad. You might be thinking that all the yumminess of food cannot possibly be explained by a mere 5 or 6 tastes. And you'd be right. Much of the sensory information we get about food comes not just from our taste buds, but also from our noses. The ability to distinguish smells is much more finely tuned than our ability to taste. When you have a cold, it's the temporary lack of smell that explains why everything tastes like styrofoam. Other senses contribute to what we call taste as well. The feel of food, known as texture, is simply a sense of touch, as we mentioned for fats. The temperature of food also contributes to its flavor. For example, the temperature of fats determines whether they arrive as solids or liquids, and this can contribute to the texture. So now that we're more aware of everything that contributes to flavor, let's look more closely at the tongue. If you put a couple of drops of blue food coloring on your tongue and look at it in a mirror, you'll clearly see little bumpy structures scattered all over its surface. These bumps are called papillae, and they are the structures that hold the taste buds. Taste buds are buried in a layer of the tongue with a small opening through which the taste molecules, or tastants, enter. Each taste bud looks a bit like a garlic bulb and may contain 50 to 100 sensory cells. Some are supporting cells, where others are cells that do the actual work of sensing different tastes. In contact with the sensory cells are the dendrites of the sensory nerves that carry the taste information to the brain. A given taste bud or garlic bulb can sense multiple tastes. That is, each clove of the bulb has one type of receptor, but different cloves have different kinds, allowing for more than one taste to be picked up in a single taste bud. An earlier notion that different regions of the tongue detect different tastes turned out to be mistaken. But how do the sensory cells sense the taste molecules? In each case, the receptors, which are transmembrane proteins, are crucial. Salty and sour tastes are the simplest. For detecting salt, sensory cells with a sodium channel known as ENaC depolarize when sodium enters them. This in turn opens voltage-gated calcium channels that initiate the action potential. As for all of the senses, an action potential sends a message to the brain, which responds by giving you the sensation, in this case, a salty taste. Likewise, sour tastes which result from acidity are sensed when the acid's hydrogen ions traverse an ion channel and enter a sensory cell. The positively charged protons cause membrane depolarization when they enter the cell and set off an action potential. Mice lacking the protein that makes this ion channel appear unable to detect sour tastes. The other three kinds of tastes, sweet, umami, and bitter, are sensed with the help of G-protein coupled receptors, or GPCRs. Remember, these are cell surface receptors that bind to signals and pass on the message with the help of G-proteins. Sweet and umami are sensed by specific pairs of GPCRs called T1Rs. Information about sweet taste is relayed by a combination of T1R2 and T1R3, while T1R1 and T1R3 work together to sense umami. In both cases, the binding of a molecule with a sweet or umami taste to the appropriate receptor results in activation of a special G protein called gusdusin. Gusdusin triggers a series of reactions in the cell that ultimately results in opening of ion channels. This triggers an action potential in the cell, which then transmits information to the dendrites of the sensory nerve. Bitter taste reception also uses the same system of GPCR signaling through gusdusin, but the receptors are much more complex than those for either sweet or umami. This is perhaps because they are important for preventing us from eating a variety of toxic foods and other undesirable substances. Humans have 43 bitter receptor genes. For tasting sweetness, by contrast, we only have 3 genes. Oddly enough, taste receptors have been found in places besides our mouths. Sweet taste receptors are found abundantly on intestinal cells. These receptors are thought to play an important role in regulating glucose balance by triggering the release of insulin and other signaling peptides when sugar is detected in the food being digested. These in turn increase production of glucose transporters for uptake of sugar following a meal. Even more bizarrely, bitter taste receptors are found in the lungs and in the airways that deliver oxygen to the lungs. Binding of bitter-tasting molecules to these receptors is actually more effective than drugs in current use for opening and relaxing of the airways. This has important implications for the treatment of asthma, emphysema, and other respiratory conditions. However, to be effective, bitter compounds must be in an aerosol that can be inhaled. Merely drinking tonic water with bitter molecules, such as quinine, will have no beneficial effect. Of course, taste and smell are closely connected, as we've already mentioned. The sense of smell, known as olfaction, is enormously important to animals, not just for the enjoyment of food, but for warning of danger and even selection of mates. It occurs as a result of binding of odorant molecules to olfactory receptor cells in the nasal cavity. Smells reach receptors by 2 routes. The primary one is via the nostrils. A second one is located where the roof of the mouth connects the throat to the nose. Chewing food releases odorants that can travel the second path to the olfactory receptors. Mucus is a viscous fluid full of glycoproteins called mucins that we met in Lecture 10. And it is mucus that covers the lining of the nose, the nasal epithelium. It serves as both a solvent for odorant molecules and an important protection against infections, since nasal neurons connect directly to your brain. Mucus is constantly being replaced with a turnover rate of about 10 minutes. Odorant receptor cells are located in the olfactory epithelium. In humans, the area of the olfactory epithelium is a little bigger than a postage stamp. Mammals have about 1,000 to 1,500 olfactory receptor genes. In humans, only about 350 of them make active receptors, but rats have almost 1,500 genes making active olfactory receptors. Perhaps that's why Remy the rat in the movie Ratatouille is such a good little chef. Each olfactory neuron expresses a single kind of receptor, but each receptor can bind several different odorants, some weakly and others more tightly. A particular odorant may bind more than one kind of receptor, again with different affinities. These combinations give humans the ability to distinguish far more than the 350 smells that we have sense cells for. In fact, a 2014 study put the number of smells humans can tell apart at 1 trillion, way more than anyone thought. The ability of olfactory receptors to distinguish different odorants is remarkable. For example, carvone is a molecule that comes in 2 stereoisomers, that is, mirror image forms that are otherwise chemically identical. The form of carvone known as the L isomer has a minty smell like spearmint. The other isomer known as the D form has the spicy aroma found in caraway seeds. Binding of an odorant by a receptor causes a G protein named GOLF or GOLF to be activated. The activated GOLF then activates adenylate cyclase, which produces cyclic AMP, as we have seen previously. The cyclic AMP produced binds to a channel with a cyclic nucleotide gate, and that releases calcium ions in the cell. Calcium in turn binds to a calcium-activated chloride channel, leading to exit of chloride from the cell and depolarization of the membrane. An action potential is generated and the signal proceeds to a central collection structure called a glomerulus. Each glomerulus contains the axons of olfactory receptor cells meeting up with the dendrites of the neurons of the olfactory bulb, a region of the brain concerned with smells. A particular glomerulus collection structure receives input only from one type of odorant receptor. Axons from as many as 25,000 receptor cells may converge to fewer than 50 neurons in the olfactory bulb. Neurons from the olfactory bulb communicate the information to the olfactory cortex, where it is processed. Unlike other sensory information, olfactory bulb information bypasses the brain's collection center at the thalamus and communicates directly with the olfactory cortex. From there, communication continues to the amygdala and the hippocampus. These 2 regions are involved with emotion and memory, explaining perhaps why smells can be so powerful in evoking memories. The sense of smell may play a role in the choice of sexual partners, even in humans. Female humans, as well as mice and fish, can apparently detect by smell a component of the immune system known as the MHC complex. And there is evidence that females prefer the smell of males with different MHC genes than their own. Children of such couples might have a more robust immune system, which would give them an evolutionary advantage. It is interesting to think about how such a system might be confounded by the modern practice of suppressing or covering up body odor with deodorants and perfumes. And of course, on Match.com or Tinder, people must rely mainly on their eyes to make their choices. Now, whether this is the best way to choose a mate or not, humans are certainly visually oriented creatures, and vision is the next sense we'll explore. The detection of light and the ability to distinguish detail by our eyes involves an amazing convergence of optics, light-sensitive proteins, nerve signaling, and brain processing. Consider that 130 million photoreceptors in the retina absorb light and transmit visual signals to the brain. The bandwidth of the human retina is about 9 megabits per second. Now, to give some perspective, standard DVDs have a bit rate that is almost the same, while a lot of video posted on the web is less than 1 megabit per second. So how exactly do we see? The anatomy of the eye is such that light passes through the cornea, pupil, and lens where it is refracted and focused onto the retina at the back of the eyeball. The retina is the location of photoreceptor cells which contain proteins called opsins that detect light. Opsins are transmembrane proteins coupled to G proteins. This is similar to the sweet taste receptors that we've seen mentioned earlier. The distinctive feature of opsins is that they hold a molecule of vitamin A that gets altered when exposed to light. Thanks to opsins, photoreceptor cells detect light and pass signals on to other neurons called ganglion cells. The axons of the ganglion cells are bundled into the optic nerve, which routes the information, ultimately, back to the visual cortex in the brain to process the signals and tell us what we are seeing. There are two types of retinal photoreceptor cells involved in light detection: rods and cones. Rods are responsible mostly for detecting light under the very low light conditions. They provide little color information since they are optimized to detect things in very dim light. What rod cells lose in color detection, they gain in sensitivity. A rod cell can detect a single photon of light. Color detection is the domain of the cone cells. Humans have three types of cone cells, each specialized to absorb wavelengths corresponding to red, green, or blue light. Cone cells operate best in bright light. Both rods and cones have opsin proteins to sense light. Slight differences in the amino acid sequence of different opsins are what determine the wavelength of light they absorb. All opsins contain a form of vitamin A known as retinal. Opsins plus retinal are known as rhodopsin. Although each type of photoreceptor cell absorbs best in a specific wavelength range, the basic mechanism of signaling is the same. In the dark, retinal and rhodopsin is found in a form called 11-cis due to the configuration of the double bond of the vitamin A at carbon number 11. When light is absorbed, the 11-cis bond reorients to the 11-trans configuration. This shape change for retinal also changes the rhodopsin holding it, resulting in activation of an intracellular G protein called transducin. The next few steps involve sodium ion channels. In the absence of a signal, these channels are kept open to allow sodium ions to move across the membrane. When activated as a result of light detection, transducin causes an enzyme called cGMP phosphodiesterase to catalyze breakdown of the molecule propping open the gated sodium ion channels and calcium ion channels. This keep the channel open molecule is called cGMP or cyclic GMP, and it's normally present in the cone and rod cells in the absence of a light signal. In other nerve signaling you have seen, it's the opening of the sodium channels that initiates the nerve signal. Here, it's the closing of the channels that starts the signal. Anyway, each detection of light by a photoreceptor leads to cyclic GMP within it being broken down by the activated cGMP phosphodiesterase, and the gates to the sodium and calcium channels close. This has the opposite effect from what you have seen before. Instead of an opening of ion gates that leads to depolarization, what we have is the closing of gates. Sodium and calcium are not able to enter, and we have hyperpolarization of the membrane. This hyperpolarization triggers downstream signaling that ultimately relays information to the brain, which assembles this information from all the photoreceptors and paints the pictures for us that amount to vision. Once a photoreceptor has fired, it goes offline. Meanwhile, neighbor photoreceptors are taking over to fire as needed. So how does the offline 11-cis retinal get back to the active all-trans form? This is handled by the visual cycle, which converts the 11-trans retinal back to the 11-cis form and reattaches it to opsins so it can be used again. Seeing color requires input from each of the red, green, and blue cone cells. Color blindness occurs if one of the three receptors is damaged or absent. Though damage to any of the sets of cones is possible, the most common type of color blindness is red-green color blindness, and it results from the lack of either a red or a green opsin. Genes encoding the opsins are on the X chromosome. Human males have only one X chromosome, while females have two. So loss of coding for an opsin on their single X is a bigger deal for men than it is for women. Consequently, 8% of males are red-green colorblind compared to only 0.5% of females. And in another blow to men, a 2016 report shows that men are almost twice as likely as women to develop hearing loss. The reason for this is not clear. Women might say it's a use it or lose it situation where men simply get out of practice and forget how to listen. Let's now consider how hearing normally works. The sense of hearing is remarkable in its range. People with normal hearing can detect sounds of pitches or frequencies between 20 and 20,000 hertz. Normal everyday speech has a mix of low and high frequency sounds, generally in the 250 to 6,000 hertz range. The length of time it takes our nervous system to respond to a sound signal is on the order of tens of microseconds. Sound detection occurs when tiny hair cells called stereocilia move on the membrane of an ear structure called the cochlea. Each of the stereocilia are attached to an adjacent one by a filament that controls the opening of an ion channel. When sound waves arrive at the cochlear fluid begins to move. The waves in the fluid move the hair cells on the membrane like surfers riding a wave. And when this happens, the up and down motion of the basilar membrane sets up side-to-side movement of the fluid between the basilar membrane and the tectorial membrane, and this causes the stereocilia to tilt. Tilting of stereocilia yanks the filament attached to the ion channel on the neighboring stereocilium and opens it, initiating the signaling process. When the brain receives these signals, it reports it as sounds. The sensitivity of the mechanism is astounding. Movements as small as half the diameter of an atom can be detected. Just like sight, the sense of hearing is dependent on ion channels. But here the more familiar depolarization of the membrane occurs as a result of opening ion channels. Depolarization leads to release of neurotransmitters that convey signals from sound detection to your brain, traveling along the cochlear nerve. Okay, and all this happens in thousandths of a second. And that's what allows you to hear everything I've said so far. Hearing loss is complex, but one thing that commonly happens is damage to the hair cells of the inner ear, whether due to age, infection, or exposure to loud noises. Damaged hair cells, of course, can't signal. Cells responding to high frequencies are located in the lower cochlea and are among the most easily damaged, thus explaining why high-frequency hearing loss is usually the first to occur. Finally, I've saved the most mysterious sense for last, the sense of touch. For example, though you might not expect it, the sense of touch is responsible for your ability to taste spicy foods. Touch stimuli can be mechanical signals where low-threshold mechanoreceptors sense contact with the skin. Low-threshold mechanoreceptors detect pressure, vibration, stretching of the skin, and movement of hair follicles in the skin. These receptors respond by triggering action potentials that communicate with sensory neurons to send information to the brain again. Thermoreceptors detect temperature signals that are lower or higher than your body's. Sometimes mechanical and temperature signals team up. Wetness, for example, is sensed by the simultaneous firing of a combination of mechanical and temperature receptors. Now you might wonder how it is that wetness sensation arises from such seemingly unrelated receptors. The answer is that the brain is where all the sensations are created from the information it receives from nerve cells. Recall that the taste of a meal is actually made by the combining information from smell receptors, taste buds, and even texture or touch. One of the prime functions of the sense of touch is to help us distinguish innocuous stimuli from harmful. Pain receptors that detect dangerous stimuli and signal the spinal cord and brain are called nociceptors, related to the word noxious. These pain receptors respond to mechanical, thermal, or chemical stimuli that are outside the range that is innocuous and alert the brain to tissue damage. Nociceptors for temperature are activated by dangerous heat or cold conditions with separate sensing cells for each. Their responses are accompanied by pain at both ends of the temperature spectrum. These are abnormal situations, and the ion channels involved signal the abnormality by opening and causing membranes to depolarize. In 2019, researchers identified a subset of neurons in the amygdala of the brain that labels a sensation as painful. Inactivation of those neurons did not interfere with the ability of mice to sense the stimulus, but they no longer behaved like they were in pain. This knowledge could lead to novel therapies for people who suffer from chronic pain. Being somewhat perverse, humans sometimes seek pain. The joys of spicy food are felt through the danger signals of nociceptors. Capsaicin, the biochemical in hot peppers that gives hotness to foods, binds and triggers a receptor called TRPV1. TRPV1 can detect high temperatures and usually signals when the temperatures reach 42 to 45 degrees centigrade, just below the threshold where heat sensors signal pain. So when you dig into your favorite spicy dish at a restaurant, receptors in your mouth tell your brain that they've encountered something dangerously hot. You aren't damaging your tissues, but capsaicin is tripping the circuits that say you are. For something activating pain circuits, capsaicin is used paradoxically in pain relief products like Therapatch and Icy Hot. Now, how it works is unclear, but it appears to interfere with pain signaling and provides a measure of relief. Our five senses allow us to function and reasonably navigate the world we live in. When it comes to some aspects of sensory perception, though, other members of the animal kingdom can pick up signals we can't, at least not very well. Bees see in the ultraviolet. Some animals sense the Earth's magnetic field. Migrating birds use magnetic sensing for navigation, and dogs may have found a new way to astonish us, with reports that they align with the north-south axis when defecating. Pit vipers use the infrared radiation to find warm-blooded creatures in the dark, and the mantis shrimp sees color better than we do, thanks to having 16 different kinds of cones. But although sensory abilities across the animal kingdom surpass us in many ways, we may catch up eventually. Scientists have already found that the brain has greater plasticity than expected. We already know that blind individuals can use touch and sound information to create a picture of their surroundings. Experimental animals have been shown to be able to process sound using the visual cortex. Meanwhile, work on synthetic retinas and advances in hearing aids are leading the way and demonstrating that our brains can learn to paint coherent pictures using new kinds of sensory input. Did you know you can see in the dimmest of lights with your rods and your cones and their retinaldehyde found in rhodopsin? It's got a bond shaped as cis, but it changes its state when a photon gets it straight. Then the signaling kicks in thanks to a transducin, 'cause its GTP ways turns on diesterase. So gated ion channels stop charges from passing through, such as sodium +1 and calcium +2.